Hi, everyone. So let's continue talking about glycolysis, this process by which cells can utilize glucose to kind of break the glucose down and try to convert some of the energy that's represented in those carbon-carbon bonds of the glucose into a form, a molecular form, that the different components of our cells, like proteins, that are enzymes, things like that, can use, which is, of course, ATP. I think it's a nice reminder that the word glycolysis, if you take it apart, the glyco part refers to sugar, right? And lysis is the breaking apart of something. So glycolysis is literally the breaking apart of sugar, in this case, glucose. We'll also talk about, at the end of today, a process by which cells can actually put together some of the leftover scraps of glycolysis to actually make more glucose molecules. And you might be surprised that some of the cells in our body can actually do this to a very limited extent. And that would be the process of gluconeogenesis. In other words, if you take it apart, gluco refers to glucose. Neogenesis means the making of glucose, right? Brand new, right? Other than relying upon our normal source of glucose, which is from our diet, right? So normally our bodies, our cells cannot make glucose as the main source of this raw fuel for all of our cells. We have to get our glucose from the food that we eat. But under certain circumstances, right, the tissues of your body have a limited capability of making glucose molecules. And so we'll talk about that today. Okay, so where we left off last time was we had learned that this process of glycolysis, right, that almost all living things on Earth do. I say almost all because there are always some exceptions, right? But nearly all eukaryotic cells, like the cells that make up our body, rely upon the use of glucose as raw fuel. We said that the first steps of breaking down the glucose through glycolysis have to be done anaerobically, meaning without the participation of oxygen. And this goes back to the ancient history of life on Earth, when life first evolved, right, in our primitive oceans, when there was no atmospheric oxygen, but there was plenty of water and carbon dioxide. So in order for life to first start, right, it wanted to take advantage of all this energy that was coming down from the sun, right? And early life needed to convert that energy somehow into molecular form, right? And so glucose was that form. Okay? So you might think, well, why did life need to actually put together glucose? If you have plenty of sun coming down, can't you just directly use that as power, as fuel? Well, but the sun always shining, right? So consider this, the very fact that our planet rotates and we have a day and a night alone dictated constraints over how life could evolve, right? Because life had to evolve in such a way that even if during daytime there's plenty of sun providing energy, it could not be that once day became night, that suddenly everything just dies, right? Because there's no sun providing energy. There had to be a way to store the energy of the sun into molecular form so that life could indeed persist, right? And so that molecular form was glucose. And so early life was already putting together glucose using the power of the sun to do photosynthesis, right? And putting together carbon dioxide and water, which there was plenty of, right? Putting in 686 kcals per mole of energy, essentially, to take those molecules and rearrange them as glucose, C6H12O6, with a leftover oxygen, right? 6 O2 molecules, right? That were then released as waste. So this glucose molecule then could be used, saved for a rainy day, so to speak, right? So when the cell needed to tap into that energy that was stored in there, it could break apart the glucose. But remember, there was very little, if no, atmospheric oxygen present. Right? So in order to get energy out of the glucose, the early life had to do it in a way that did not require or depend upon oxygen. And that was a big limitation. Because remember, in our previous lecture, I showed you this nice cartoon showing that if you imagined a glucose molecule, right, being busted apart, if you had a lot of oxygen molecules available, 6 O2 molecules, and you busted those apart, then those oxygens, right, because they're very greedy for electrons, would happily oxidize the glucose, right, allowing for the electrons that are part of the carbons in the glucose molecule to now engage with oxygens. So you'd form carbon dioxide, CO2, and you'd form leftover water. And that's how you maximize getting all of the bond energy out of a glucose molecule. In other words, that's how you would release the 686 kcals per mole that were first put into making that glucose in the first place. But without oxygen, right, you can't do that. Right? So for the first billion years of life on Earth, life had to take apart the glucose in a way that wasn't very efficient without oxygen available. And that's what glycolysis was, right? So glycolysis alone is highly inefficient for extracting energy. We learned last time that glycolysis happens in 3 phases, right? Or our textbook calls it 3 phases, other places will call it 3 stages, etc. But mainly the first stage in this cartoon here is where you take the glucose and you have to rearrange it a little bit, the bonds, to make a symmetrical molecule. You end up putting 2 phosphate groups onto that by using up 2 ATPs that the cell already has in it. Right? So 2 of those phosphates get added, and then you split the glucose into 2 identical half molecules, each one of which is 3 carbons hanging onto one of those phosphates that came from one of the 2 ATPs. Right? Remember, phase 2 and phase 3 is everything is times 2 here, because each one of these represents half of the original glucose. Right? Then you're going to have another phosphate group that gets added onto this during phase 2. This cartoon doesn't show the other phosphate group added, but imagine another orange P being added onto this here. And then you proceed to go ahead and take those phosphates off, add them to ADP, adenosine diphosphate, and make a third phosphate added onto the ADP to make ATP. Right? And in the process, right, you make an ATP molecule, and the electrons that are being removed from this, in other words, oxidized from this, right, have to go somewhere. So remember, big proteins functioning as enzymes are handling this. Right? They recognize and bind to this particular shape of this intermediate molecule, right? And they do the oxidation. They remove a pair of electrons. Those enzymes don't want to get stuck with those electrons, because if they got stuck with them, then they're not going to work again until they somehow can unload those electrons, right? So a more convenient way of dealing with this is you utilize a coenzyme, something like NAD+, which we can think of as an empty basket that's ready to take electrons. So the enzyme would pull electrons off of this 3-carbon intermediate, dump them into NAD+, turning that basket into a filled NADH basket that then can carry those electrons away. And therefore, the enzyme is free not to worry about being burdened by carrying electrons on its own. Right? Then in phase 3, right, the second phosphate that got added in phase 2 ends up contributing to the production of another molecule of ATP. Right? Leaving you at the end of phase 3 with pyruvate, right? 3-carbon chain that is the result of glycolysis. And in the process, you've made 2 ATP molecules here times 2, because remember, this is actually happening in parallel, right? But since you had to invest 2 ATPs to start this process, right, your net gain is only 2 ATPs. Right? So in other words, the net yield of 2 ATPs from the glycolysis of one molecule of glucose really only represents about 3% of the total energy in that glucose molecule, right? So it's really inefficient because most of the energy is still locked up in these pyruvate molecules, in these carbon-carbon bonds that the original photosynthetic microbe or plant, right, had invested energy from the sun to make these bonds in the first place. Right? And that's because beyond this point, without oxygen, you can't really go any further with the breakdown of the pyruvate. Right? So that's why we learned at the end of last lecture that anaerobic microbes, certain bacteria, even certain types of simple eukaryotic cells like yeasts and things like that, right? They do glycolysis, they end up with pyruvate, but at least with the pyruvate, they can use the pyruvate in the process of fermentation. Right? Why? Is it to get more ATPs out of the pyruvate? No, because again, without the presence of oxygen, you're not going to be able to oxidize the pyruvate further. But what you can do is you can take the electrons that are in these NAD-filled baskets of NADH, right? And you can empty those baskets to dump the electrons back onto pyruvate, thereby freeing up those baskets to go back and contribute in glycolysis to keep the glycolysis process going, right? So the fate of this pyruvate depends on different factors, the kind of organism you're talking about, the specific cell type, and importantly, whether you have O2 available, right, to maybe be able to oxidize the pyruvate further. So consider this, right? In anaerobic fermentation, if you don't have oxygen available, at least the pyruvate can go through fermentation as a way of kind of freeing up some of those electron-carrying baskets, these NADHs, right? But if you have oxygen available, right, there is another route through which you can send the pyruvate, right? Because I said if the pyruvate still represents the majority of the energy in the glucose molecule, if you have oxygen around, then you'd probably much rather use that oxygen to fully oxidize the pyruvate molecule to get that energy out. So that is aerobic respiration. And that's what we will talk about starting with next lecture. Okay. But for today, I wanted to finish our discussion about glycolysis and the process of fermentation that gets linked on after pyruvate. Yeah. So we introduced the two main fermentation pathways last time. So let's kind of talk about them in a little bit more detail. Remember that what we are talking about here is starting from glucose and going through the three phases of glycolysis. An anaerobic microbe, like a bacteria or a yeast, something like that, can produce 2 ATP molecules as the net production of ATP out of glycolysis. Right? And in the process of making these ATPs, it's going to fill these NAD+ empty baskets that carry electrons away, turning them into NADH. Right? And the end result of glycolysis is you have this pyruvate molecule, right? Essentially representing half of the glucose, half of the starting glucose, right? These 3 carbons here. So we said that anaerobic microbes can take this pyruvate and at least use it as a place to dump those electrons out of these filled baskets. Okay. So here you have an enzyme, lactate dehydrogenase, or LDH, that will work on the pyruvate. And it will take the filled NADH basket, right, that has a pair of electrons in it, plus you will have protons floating around, right? And it'll put that pair of electrons and pair of protons onto the pyruvate, thereby making lactate. Right? So look at the structure of lactate here and compare it to the pyruvate. Right? What do you see that's different? Well, pretty much the only thing that's different is you have these 2 additional hydrogens on it, right? And those 2 additional hydrogens represent proton plus an electron, right? So if you had 2 electrons in the NADH that want to be given up, right, one of those electrons will pair up with one of these protons here, this H, and get added to make the lactate, maybe this one here, right? And the other one, the other electron will pair up with a proton that's pulled in, right? And maybe that's this hydrogen over here, right? So now from pyruvate, you've made lactate. And in the process, you've emptied this basket so that the NAD+ empty basket can go all the way back and help keep glycolysis going. Right? As far as the microbe is concerned, the lactate really is the end of the line now, right? So lactate is the waste product of a microbe going through glycolysis and also doing fermentation to free up the coenzyme electron carrier. And so lactate, is what gives things like dairy products like cheese, yogurts, that nice tanginess that we love so much. Right? So many anaerobic microbes can start with glucose for glycolysis, or they can start with other disaccharides. Remember, glucose is a single monosaccharide, but we learned earlier in the semester, oftentimes you have glucose paired up with another monosaccharide to make a disaccharide. For example, a glucose and a galactose together is what lactose is, right? So of course, lactose is milk sugar. So many anaerobic microbes will start with glucose or lactose, right, to go through the process of glycolysis. And so we have learned, of course, to take use of this or make use of this to give things like cheeses and yogurt that nice tangy flavor, which is really just the lactate. Okay. I want to point out something to you. Notice the name of this enzyme, the lactate dehydrogenase, right? The LDH. We just talked about how this enzyme is taking a pair of electrons and a pair of protons, emptying out this NADH basket, sticking that pair of electrons and protons onto the pyruvate to make lactate. And you're like, wait a minute, wouldn't that process of adding these H's onto the pyruvate to make lactate, wouldn't that be hydrogenation, right? So why is it called lactate dehydrogenase? That almost makes it sound that it works in the opposite direction, right? It's an enzyme that works on lactate and takes protons or hydrogens off of it, right? That sounds like a dehydrogenase. Right? Why would it be named that way? Is it a big mistake? No, because as it turns out, many enzymes, right, can work in both directions. In other words, in this case, the LDH enzyme can take electrons and protons and stick them onto pyruvate to make lactate, or I guess it's this direction in the figure, but it can also grab lactate and take those electrons off to drive it in the opposite direction. And it just turns out that historically, right, when this enzyme was first described, it was first recognized for that function going in that direction. So it was named lactate dehydrogenase, right? And the name just stuck, right? So that happens with a lot of enzymes, right? So it's kind of this weird thing where historically it just reflects how they were first described, even though we have a much better understanding now of different functions. I mentioned that in our bodies, we have the ability to actually do lactate fermentation. So why would we want to do that? Well, think about this. If you have certain cells that are starved for oxygen, right, without enough oxygen, you can't fully oxidize the glucose. Right? So what are you left with? Well, you're left with pyruvate. Okay. And if without any other choice, what are you going to do? You can't just keep building up pyruvate in your cells, so you can do fermentation. Okay? So muscles perform fermentation when the oxygen is used up too quickly. For example, if you're working out really hard, right, and you're really driving your skeletal muscles hard, they may actually be starved for oxygen, right? They're not getting enough oxygen to fully make use of the glucose. And you better believe those muscle cells, right, are metabolizing glucose, right, at an incredible rate. because they need fuel. They need to convert lots of glucose into ATPs to keep them functioning, right? If you're running really hard, working out really hard, right? Without enough oxygen, they will just go through fermentation to make lactate. And that lactate then builds up in our muscles, right? It gets released into our blood. And many times that buildup of lactate or lactic acid is what is credited for things like sore muscles, right? Sore, aching muscles after you work out is believed to be, at least in part, your nerves picking up on the fact that there's this lactic acid there. Right? So we say that if cells are starved for oxygen, they are experiencing hypoxic conditions, right? Low oxygen conditions. So let's look here. So the glucose in a muscle cell, right? It is going through glycolysis, and it only gets 2 crummy ATPs out of glycolysis, and it makes pyruvate. Right? Normally, because we have available oxygen, this pyruvate doesn't have to be funneled down a fermentation pathway, because our cells would much rather still work on getting energy out of the carbon-carbon bonds in the pyruvate if oxygen is available, right, through aerobic respiration, which, like I said, we'll talk about next time. But if there is no oxygen or not enough, then the cell will send the pyruvate through a lactate fermentation pathway. Right? Part of the reason we evolved to do this is because that lactate that then gets into your blood can go to places like your liver, right, or places like your heart, where that lactate can actually be converted back into glucose. Right? So this asterisk says lactate is removed from the muscle through the blood and sent to the liver, where it is used as the starting point for making more glucose through a process that we call gluconeogenesis, right? Which we will talk about at the end of this lecture. This might sound kind of magical to you because you're like, wait a minute, you started with a molecule of glucose, you've used it to get a couple ATPs out of it. And what you're saying is when you end up with lactate, you can still make another molecule of glucose. That sounds like magic, right? That's like getting energy out of nothing. It's not true. And in the last slide of today's lecture, we'll discuss why. Other examples of cells in our body that might function in these anaerobic types of conditions are things like diseased cells, like cancer cells. So if you have a cancerous tumor that's forming and you have cells that are dividing out of control, right, and forming a massive tumor, that growing tumor will push the available blood supply of all the little arteries and capillaries, right, farther and farther out from the cells that are in the middle of the tumor. As a result, those cells in the middle of the tumor start being starved for oxygen, right? They are experiencing hypoxic conditions. Now, cancer cells, even though they are really messed up cells, right, because of lots of mutations and lots of things going out of control, I mean, they're still living cells, right? And so one of the most fundamental things that a living cell needs is ATP in order to function, right? So these cancer cells that are in the middle of a big growing tumor are really stressed out, and many of them just die, right? And so as a result, oftentimes large tumors are really kind of nasty on the inside. There's a lot of necrotic tissue, right, from tissue dying in the middle. However, some cancer cells, through random mutations underneath this kind of natural selection pressure for them to favor mutations, right, to get around this, can end up having mutations that increase the amount of glycolysis that they do. Okay? So we learned that when cells are taking in glucose from the outside, the glucose cannot just float through the membrane. The glucose has to be transported in, right, through something like the GLUT1 transporter protein. So here you have the GLUT1, this protein, this purple and blue protein that's in your cell membrane, which is the gray thing here, right? And here's a glucose molecule, right? And the glucose molecule from the outside binds the GLUT1 transporter, and the GLUT1 transporter changes its shape so that the glucose is now on the inside. Okay? So one of the mutations that we often see in cancer cells is that cancer cells really, really increase the expression of the GLUT1 gene that makes the GLUT1 transporter protein. Right? The glycolysis itself of the glucose is still limited to only making 2 crummy ATPs per glucose molecule. But if you can get a ton of glucose into the cell, way more than normal, then at least those 2 ATPs per glucose begin to add up. Right? Let's look at this. So here is some cool pathology stains looking at some tumor sections. So here in this early tumor, when the tumor was first detected in the patient and maybe they took a biopsy, what they've done here is they've stained the cells with antibody that detect the GLUT1 protein on the surface of the cells, right? And makes it appear brown in this slide here. So you can see these would be more like normal cells on the either sides, right? You don't see a lot of GLUT1 on their surface. I mean, they have it, obviously, but not enough for you to get a nice strong brown signal in here. But look at the cancer cells, right? They're beginning to get more and more brown because they have mutated to make much more GLUT1 transporter on their outside. So that's in the early tumor. As the tumor grows, right, you would imagine that the cells that are getting more and more in hypoxic conditions, starved for oxygen, right, would favor mutations that make even more GLUT1. So on the right, let's look at what a section looks like in a late tumor. Look at that, right? So again, here are the advanced aggressive tumor cells being stained for GLUT1 on their surfaces compared to over here, normal cells where you can barely detect GLUT1. Right? So this is known as the Warburg effect. And for those of you who are interested in learning about this in cancer, especially those of you who are in the MCB program, when you take the biology of cancer class, we look at mutations like this. Right? But for our Bio 275 class, I just wanted to use that as an example, right, to get you thinking about how cells need to be able to regulate things like glycolysis. And this is another good example of what we talked about, gene regulation, right? These tumor cells have mutated in such a way that the gene regulation of the GLUT1 gene making normal amounts of GLUT1 transporter on their surface, right, has been tweaked and messed up to actually make a lot of GLUT1. So lactate fermentation is one form of fermentation. The other key fermentation pathway is alcohol or alcoholic fermentation. All right, so again, microbes can start with glucose. Right? So this glucose might come from things like the sucrose that's in fruits, right? So sucrose is glucose and fructose together, right, as a disaccharide. Or this glucose might come from maltose, right, from different grains. So maltose is actually a disaccharide of 2 glucose molecules put together. It's just 2 glucoses together. Right? So those microbes would have ways of breaking those disaccharides apart to get to the glucose. And again, these microbes would use those glucose molecules through glycolysis. They'd make their 2 crummy ATPs out of it. They'd fill one of these NAD+ coenzyme baskets with pairs of electrons, right? And they end up with pyruvate. So they may have enzymes that can convert the pyruvate into acetaldehyde, right? So you can see here, here's acetaldehyde. So now the acetaldehyde only has 2 carbons in it. So what happened to the 3rd carbon in the pyruvate? Well, it's released as carbon dioxide. There goes the 3rd carbon, right, as CO2 that's released. And then the microbes would have another enzyme, alcohol dehydrogenase, ADH, that would take the acetaldehyde, right, and use that as the dumping place for those electrons that were being carried by the NADH baskets, right? And they take a pair of electrons with 2 protons, stick it on the acetaldehyde, and you've now made ethanol, right? So again, the ethanol is the final waste product of glycolysis, right? So consider this. The most microbes or many microbes don't just choose one or the other. They don't just do lactate fermentation or just do ethanol fermentation. A lot of times it's a combination of both because they have all the enzymes to do it. So that combination of, for example, fermentation releasing CO2 gases is what gives bread dough, it's nice rise, right? So when the yeast is breaking down the sugars that are in the wheat, for example, right, and the sugar maybe that you add to the dough, right, as you let the dough proof or rise, what's causing it to puff up? It's the carbon dioxide that's being released through fermentation, right? It's kind of neat. That means your microbes they put in there, the yeast, right, they're alive. They're doing glycolysis to make ATP, and then they're tacking onto it fermentation, and that releases CO2. They may also be doing lactate fermentation, right? In which case you get nice sourdough bread, right? That is nice and rising because of the CO2, but also nice and kind of tangy because of the lactate, right? So you say, but wait, how does the alcohol come into this then? Because you don't end up with bread that tastes like alcohol, right? No, right? Because you bake the bread, so you essentially kill the yeast and you stop it before there's enough ethanol buildup, right? But if you just had dough and you never baked it, right, and you just let it sit there with some water, what would happen? It would ferment all the way to ethanol, right? And so essentially, it would become like the mash or the type of stuff that you'd use to make alcoholic beverages, right? So wine, of course, is grapes that are mixed with certain microbes, and they can take this process all the way to ethanol, right? Again, notice alcohol dehydrogenase. Where have we seen that before? We saw that when I was giving you the example of how we metabolize ethanol, right? When we drink ethanol, our body has to break it down, otherwise it poisons us, right? We use alcohol dehydrogenase as the enzyme that takes ethanol and breaks it down to acetaldehyde. So in other words, another example of how this particular enzyme can do things in two directions, right? In this case, if there's a lot of acetaldehyde, like in microbes working on sugar, making pyruvate, turning it into acetaldehyde, the direction will favor converting it towards ethanol as a waste product, right? In our bodies, if we drink a lot of ethanol, right, the enzyme will work in the other direction, helping break down the ethanol to acetaldehyde. Then you say, wait, does that mean that we end up also going all the way and make pyruvate out of the ethanol? No, because you'll remember in the previous lecture where I showed you that series of how we metabolize beer eventually down to CO2 and water, we have another enzyme, an aldehyde dehydrogenase that would then come in here at this step, right, and take this pathway in a different direction, right? Instead of going up to pyruvate, the aldehyde dehydrogenase that we have would take this acetaldehyde and break it down in another direction that leads towards eventually carbon dioxide and water in our bodies, right? Either way, I think this is kind of interesting to think that these processes were invented by microbes billions of years ago, right? Like life, started around 3.5 billion years ago. And for the first billion years, all life did was anaerobic respiration, right? Converting glucose into pyruvate, and then maybe sending pyruvate through fermentation to keep the glycolysis going, right? So here we are, 3.5 billion years later, we've evolved as sort of like the supreme dominant species of the planet, right? And what do? Hey, we've learned how to eat and drink the poop of the microbes, right? The lactate and the ethanol. Yeah, so we're really not that smart, right? Okay. So in our bodies, we also learned that if we eat a lot of sugars and we have a lot of extra sugars left over, we don't want to just waste it, right? Because you never know evolutionarily, right, when your next meal is going to be. So you want to hang on to those sugars. So one convenient way of hanging on to those sugars is stringing glucose together into things like glycogen, right? So mammals like us, or this little mouse, right, in our livers, we string together glucose molecules to make this nice glycogen molecule. Plants also do this. Plants that are busy doing photosynthesis to make their glucose, right, if they don't need that glucose right away, they need to store it, save it for a rainy day or nighttime when there's no sun, right? Otherwise, those plant cells would quite literally die if they didn't have that energy stored up. So that glucose gets chained together in plants as starch. And we learned that our bodies have enzymes that are the right shape to be able to tap into this. So if you have a long polysaccharide of starch or glycogen, right, and your body needs to get some more sugar flowing around in your blood, let's say it's been 6 or 8 hours since you had your last meal, right, and your tissues in your body are beginning to run low on sugar to power them, no sweat. You have glycogen stored up in your liver, right? So you're going to have enzymes that then start breaking the glucose molecules off. So the way they'll break them off is you'll have an enzyme such as a starch or glycogen phosphorylase, right, that can take a phosphate group and add it to the C1 position of one of these glucose molecules, thereby disconnecting that glucose molecule from the glycogen, for example, and now you end up with this free glucose 1-phosphate, right? It's a glucose molecule with a phosphate group at the 1 position, the carbon 1. All right. So how does this all fit in with our discussion of glycolysis? This figure, although it looks really scary busy, is really not that bad. In this kind of peach color shaded area is just phase 1 of glycolysis. Remember, glycolysis is 3 phases. Phase 1 is just about taking a monosaccharide like glucose, right, rearranging it a little bit, and eventually, right, you're going to add phosphate groups onto it by spending 2 ATPs. And then by the end of it, you're going to end up with glyceraldehyde 3-phosphate over here, right? But what this figure shows us is that you don't always necessarily have to start with glucose, right? In the previous slide, I mentioned that, for example, microbes, they can use glucose straight up, right? And that therefore the glucose would just start here in the figure. But many sugars that are available are disaccharides, combinations of glucose with something else. So maltose, like I said, is just 2 glucoses together. Sucrose is a glucose with a fructose, right? Lactose, like in milk, is a glucose with a galactose. Right? So just as long as you have enzymes available, like this E8, E9, E10, you don't have to worry about what these things are called, right? They're just mentioning to you that there are different enzymes here in this figure, each one of which takes care of breaking down this disaccharide to get the glucose out of it, right? The other monosaccharide, like for example, the lactose, when it gets broken down by this enzyme that this is just labeled E8 here, right? The glucose part of it, of course, would then become this glucose here that's feeding into glycolysis right there. But the other galactose that came off the lactose, it can also be put to use because after all, it is just another 6-carbon sugar, right? It'd be a waste not to use it. So it would have a series of enzymes that step by step by step turn the galactose into glucose 1-phosphate, right? And that would be the same glucose 1-phosphate that is the glucose 1-phosphate that comes off of, let's say, the glycogen or starch, right? When we decide to break off a glucose from there, you end up with glucose 1-phosphate, and then that can be modified. So the phosphate on the glucose 1-phosphate can be rearranged and the phosphate can be put on the 6th position. And that means it could join the glycolysis pathway here as glucose 6-phosphate, right? The same glucose 6-phosphate that you'd end up with if you started with glucose and spent your first of 2 ATPs to make the glucose 6-phosphate here, right? So then fructose, for example, right, that comes from the breakdown of sucrose into glucose and fructose. Similarly, the fructose can enter glycolysis over here. Right? So you have a fructose molecule, you break a molecule of ATP to take one of those phosphates off of it and stick it onto the fructose, and you've now made fructose 6-phosphate. Right? So you're entering the glycolysis pathway here. Right? So all of these steps over here, then you would then have to break a second ATP, right? Your second investment in an ATP, right? To make the fructose 1,6-biphosphate that then gets split in half to make the 2 3-carbon molecules at the end of phase 1 of glycolysis, right? 1 glyceraldehyde 3-phosphate comes from, let's just call it the bottom half of the fructose being cut in half. right? And this other molecule up here, the hydroxyl acetone, right, is like the top half of the fructose molecule, right? But that can be converted by another enzyme into glyceraldehyde 3-phosphate. So that's why we say, for convenience sake, the end of phase 1 of glycolysis is you've now made 2 G3Ps that feed into stage 2. Notice also, what do we have up here? We have glycerol, the same glycerol that's used to hold things like fatty acid chains together to make our cell membranes, right? That's a 3-carbon molecule. Can that be used for energy? Yeah, sure. So the glycerol can be converted, right, eventually into glyceraldehyde 3-phosphate, right, to also enter the glycolysis pathway there. Why would you do that? Well, if you don't have enough sugar, right, you have to get 3-carbon molecules from other sources. So you might have to start breaking down things like glycerol. You might have to start breaking down certain proteins, right, in order to get the raw fuel to enter glycolysis. The last thing I want to point out is this. I think many of you who are maybe very astute and observative, is that even a word? Observant, right, would comment that, look, if you break a glucose off of glycogen, right, and make glucose 1-phosphate, and it feeds into glycolysis over here, right, doesn't that mean you've essentially bypassed this first ATP that needs to be spent if you were starting from a free glucose originally, right? And wouldn't that mean then that if you take glucose from glycogen, that you only invest 1 ATP, but at the end you make 4, so 4 minus 1 is you actually get 3 ATPs? Wouldn't that be way better, right? No, that's sort of a false kind of commodity. Why? Because we forget that in order for the glycogen to be put together in the first place by enzymes using energy to add glucose together, right, that was your other investment of an ATP, right? To even make the glycogen in the first place requires an ATP per glucose that you link on, right? So it still ends up being that you've spent 2 ATPs in order to get into this part of glycolysis, right? So you don't actually somehow magically gain something by using the glycogen in your body. All right. So all of these steps require enzymes. And we said that these enzymes are big proteins that evolve to be just the right shape so that they can bind to specific substrates. And those substrates would be all the different intermediate carbon or intermediate organic metabolites of the glucose as it's being broken down and manipulated, right? Each step of the way, right, the molecule is a slightly different shape. You would need a different enzyme that evolved to recognize that particular shape. Right? We learned in our discussion of enzymes that enzymes work by lowering the energy of activation so that a bond breaking or forming that could naturally happen, right, but normally might take tens of thousands or millions of years to happen on its own, right, which would be not compatible with life. Right? If you have an enzyme there, right, an enzyme can speed that up dramatically. So something that would normally take 10,000 years if you just sat there waiting for these 2 bonds to form might take, you know, a millisecond or less if you have the right enzyme working on it. So we said that an enzyme then works by binding to substrates, forming an intermediate enzyme plus substrate form, and then you end up doing something, catalyzing the substrate, and releasing products, right? We said now that these can work in both directions. Just like you could say this enzyme here could work on this blue substrate, right, and release 2 different products, the purple and the yellow, from it. Likewise, this enzyme could conceivably bind a purple molecule, a yellow molecule, put them together to make a blue molecule, right? So that's why we say this thing can go in either direction. If no extra energy is required for the enzyme to do this, right? If this process is just a result of the enzyme being the right shape to hang on to these 2 potential substrates or 2 products, right? And that is enough to greatly accelerate the formation or the breaking of the bond between them, then the enzymes can happen in both directions. So when it wouldn't work is if, for example, right. So just to illustrate, remember, an enzyme like this one up here, this big green thing, is most likely a pretty complicated protein, right? So if you imagined unraveling that protein, right? So here's my little, what is this? My little toy, my kid's toy that I've shown you before, right? Think about it as one long polypeptide chain, right? So each one of these little links along the way is a different amino acid, right? And so maybe this is the N-terminal end of the protein, maybe this is the C-terminal end, right? And this is all coded for by the gene in your genome that has the right order of DNA bases that can be transcribed by RNA polymerase to make an RNA molecule. That RNA molecule gets edited, right, to make a messenger RNA. And then that messenger RNA in your cytoplasm gets read by the ribosome. And the ribosome then puts together individual amino acids that are being carried in there by tRNAs, right? And if the tRNA matches the next triplet codon in the mRNA, right, that really reflects what was in the DNA to start with, then you chain together this particular set of amino acids, right? And we said that the protein would fold up into our functional three-dimensional shape, and we learned that maybe some of these amino acid residues that are normally far apart from each other in the linear unraveled proteins, so we're talking about the orange, green, and yellow ones here, like this section here on the left and this section here on the right, once it's all folded up, those might actually be, right, part of the same activation or binding pocket for substrate. So here it's working on this molecule here, and it's just the right shape to fit this molecule. And by binding it, it facilitates or accelerates the breaking apart of that molecule. But this won't work if energy is required in order for the enzyme to do this. If the enzyme has to be the right shape to bind an ATP and break an ATP in order for it to change its shape to make the products in the first place, I think you can already intuitively realize then that this enzyme can't work backwards, right? So in this case, the arrow moving from left to right is huge, whereas the arrow moving from right to left is either small or non-existent. Right? So let's look at that in these illustrations of glycolysis. Right? So this is phase one of glycolysis again, starting with glucose on the left, ending up with two molecules of G3P or glyceraldehyde 3-phosphate. And you have a series of enzymes, right? Enzyme 1, 2, 3, 4, that each take their turn working on this intermediate metabolite of the glucose, right? Notice now some of these enzymes, like number 2 over here, can do this reaction in either direction, whereas other enzymes, the ones that require the breaking of ATP, can only do the reaction from left to right here, right? So in other words, if you were to try to reverse this, right, and start from one of the downstream metabolic products of glucose and reverse this using enzymes to get back to glucose, right, you'd have a problem here at these two steps where the arrow is really big and only pointing to the right. But why would you want to reverse this anyhow, right? Why would you want to reverse glycolysis? Well, again, there are certain times physiologically when your tissues are starved for glucose, right? And maybe you don't have enough available glucose to supply them. You got to come up with something, right? And so that something might be taking the scrap leftovers of glycolysis, for example, and reassembling them into another glucose molecule, right? So if glucose supplies are running low. So this is the last slide for today, and that's what this is. Gluconeogenesis is essentially reversing glycolysis so that cells can make glucose from downstream metabolic processes. If we look on the left, this is stage 1 of glycolysis, starting with glucose, and you go down, you end up with 2 molecules of glyceraldehyde 3-phosphate, right? And these are the enzymes 1, 2, 3, 4, 5 that participate, right? We said 2 of those enzyme steps, because they break ATP, right, they can only go in one direction. Whereas other steps, like the second step here, right, involving enzyme 2, it can go either the blue direction this way or the orange direction that way. Right? So that means like gluconeogenesis, right, is going to require different enzymes at certain steps, right, to compensate for the fact that the original enzyme involved in glycolysis cannot catabolize the reaction in the opposite direction. Right? And this illustration on the right just shows you. For example, skeletal muscle, if you're using a lot of glucose, right, and you go through glycolysis and you end up making lactate, lactic acid in your muscles, that lactic acid can go through your blood, and in your liver can take that lactate and put together glucose molecules again, right, through this process of going backwards, right, and you end up with glucose, right? Now you might say, why? I still don't get it, because it still looks like magic, right? Because then the implication is this orange glucose over here now can go and magically provide more ATPs. No, right? Because look, what does it take to drive glycolysis backwards, right? Because you have those steps along the way that require different enzymes, you actually have to invest 6 molecules of ATP to go from lactate all the way back to glucose, right? That's actually less ATP. I mean, that's actually more ATP than a glucose molecule would give you to start with through glycolysis, right? So you say, why then do our cells do this? Because there are certain tissues in your body where it is hyper critical that they have enough glucose. For example, your brain, right? If you are starved for glucose, you know, it doesn't really matter if the cells at the ends of your fingers or your toes get plenty of glucose to keep surviving, right? But it does matter that the cells in your brain get enough glucose to keep surviving, right? So under stressful conditions, those few molecules of glucose that are made by the liver get sent to those tissues that need glucose the most, right? And that's why we evolved gluconeogenesis. Right? The thing I want to leave you with, I know we're out of time, just a thought question, right? We said that glycolysis evolved because early life had no other choice but to come up with some way of taking the glucose that they've made using the energy of the sun and the plentiful carbon dioxide and water, right? Making glucose in order to now do something with that glucose to make ATP without any oxygen. The best that life could do, evolving evolution-wise, is glycolysis, right? Yet I've also implied, right, that all of the cells in our body still start off by doing glycolysis, right, on glucose, even though we have tons of available oxygen, right? Why did we evolve that way, right? Why not just start from scratch and just say, now that we have tons of oxygen in the air, we don't have to go through this very inefficient glycolysis pathway? Right? So I want you to think about that, and we'll answer that question next time. Okay? All right. So let's go to the chat. And if there's any questions, I'm happy to try to answer them. Okay. Looks like there are no questions today. All right, that's fine. All right, so next time we'll start talking about aerobic respiration. In other words, what our cells can do now that we have plenty of oxygen, right? Starting from glycolysis and building up on that. All right, have a good rest of your day. I'll see you on Friday. All right, bye-bye.